Hello and welcome to Unit 1.2, Carbohydrates. Carbohydrates are polymers, or more specifically they are polysaccharides. Monosaccharides are the monomers from which larger carbohydrates are made. Glucose, galactose and fructose are common monosaccharides. Disaccharides are formed by the condensation of two monosaccharides. A condensation reaction between two monosaccharides forms a glycosidic bond. Maltose is a disaccharide formed by condensation of two glucose molecules. Sucrose is a disaccharide formed by condensation of a glucose molecule and a fructose molecule. Lactose is a disaccharide formed by condensation of a glucose molecule and a galactose molecule. You need to know the structure of the three main monosaccharides, glucose, fructose, and galactose. Learn to draw these in preparation of your exams. More importantly, you also need to know the isomers of glucose, alpha and beta glucose. Note that the groups on the right are reversed. You can see this is the diagram now on screen. Now, let's talk about the polysaccharides which these monosaccharides form in more detail. Cellulose is a polymer of beta-glucose. It's made of long, unbranched chains of beta-glucose. Condensation reactions link carbon atom 1 to carbon atom 4 on the next beta-glucose. The glucose subunits in the chain are oriented alternately upwards and downwards. The consequence of this is that the cellulose molecule is a straight chain rather than curved. The cellulose chains are linked together by hydrogen bonds to form strong fibers called cellulose microfibrils. Because of those fibers, the tensile strength of cellulose provides structural support for the cells, e.g. the cell walls, and prevents plant cells from bursting, even under very high water pressure. Another polysaccharide is starch. Cells get their energy from glucose. Plants store excess glucose as starch. When a plant needs more glucose for energy, it will break down that starch to release the glucose. Starch is a mixture of two polysaccharides of alpha-glucose, amylose and amylopectin. Amylose is a long, unbranched chain of alpha-glucose. The angle of the glycosidic bonds give it a coiled, cylinder-like structure. This compact structure is good for storage because more glucose can be stored in a small space. Condensation reactions link carbon atom 1 to carbon atom 4 on the next alpha-glucose. Amylopectin is a long, branched chain of alpha-glucose. It has side branches which allow the enzymes that break the molecule down to access glycosidic bonds easily, meaning the glucose can be released quickly. Since starch is insoluble, water cannot enter the cells by osmosis, which makes it very good for storage. All the glucose molecules in starch can be orientated in the same way. The consequence of this is that the starch molecule is curved, rather than straight, and the size of the molecule is not fixed. The final carbohydrate you must know about is glycogen, Glycogen. Glycogen is the main energy storage material in animals and is made by some fungi too. It's a polymer made from repeating glucose subunits and varies in size, typically consisting of 30,000 units. Animals store excess glucose as glycogen, another polysaccharide of alpha-glucose. In humans, it is stored in the liver and some muscles. It's used in cells where large stores of dissolved glucose would cause osmotic problems. Glycogen has a similar structure to amylopectin, but it has a lot more side branches. These extra branches mean the stored glucose can be released very quickly, quicker than starch can be hydrolyzed. This is something that is very important in animals due to the fact that muscles contraction constantly takes up a lot of energy. As a result of the branches, glycogen is very compact, which makes it very good for energy storage. Glycogen also does not affect the osmotic balance of cells. It doesn't cause too much water to enter them. Finally, you must know about how to test for sugars. Sugar is a general term used to describe monosaccharides and disaccharides. All sugars can be classified as either reducing or non-reducing. Reducing sugars include all monosaccharides, such as glucose, and some disaccharides, maltose and lactose. Note that the disaccharide sucrose is not a reducing sugar. Here is how to test for reducing sugars. Add Benedict's reagent, which is blue, to a sample and heat it in a water bath that boils the mixture for 5 minutes. If the test is positive, it will form a colored precipitate, solid particles suspended in the solution. The higher the concentration of reducing sugar, the further the change of color goes. If there is no reducing sugar, the solution will remain blue. If there is reducing sugar, it will turn green, then yellow, then orange and then red. Red means there is a high concentration of reducing sugars in the solution. You can use this to compare the amount of reducing sugars in different solutions. This is called the Benedict's test. The test can be described as qualitative because it produces a color change from blue to green to yellow to orange to brick red. The qualitative test is also regarded as semi-quantitative as the color obtained correlates to the concentration of reducing sugars in the solution. See observations below. This allows for a rough estimation of the amount of reducing sugar present, but you cannot know the exact amount and therefore it is not quantitative. A more accurate way, though, is to filter the solution and weigh the precipitate. In this way you could produce quantitative results. If the result of reducing sugars is negative, there may still be non-reducing sugar present in the solution. To test for a non-reducing sugar, like sucrose, you first need to break them down into monosaccharides. Take a fresh sample of the solution and either heat it with diluted hydrochloric acid or hydrolyze using enzymes. You can then neutralize it with sodium hydrogen carbonate. Once you have done these, you can continue the Benedict's test in the same way you would for a reducing sugar. If the test is positive, it will form the colored precipitate just the same as for a reducing sugar. This is because the sucrose has broken down into glucose and fructose, and all monosaccharides are reducing sugars. If the test is negative and the solution stays blue, and it means the solution does not contain any sugar, either reducing or non-reducing. How do you test for starch? The test is very simple. Just add iodine dissolved in potassium iodide solution to the test sample. If there is starch present, the sample changes from brown-orange to a dark blue-black color. In your exams, make sure you always talk about iodine dissolved in potassium iodide and not just iodine alone. To recap, monosaccharides are the monomers from which larger carbohydrates are made. Glucose, galactose and fructose are common monosaccharides. Disaccharides are formed by the condensation of two monosaccharides. Glycosidic bonds are formed. Maltose is a disaccharide formed by condensation of two glucose molecules. Sucrose is a disaccharide formed by condensation of a glucose molecule and a fructose molecule. Lactose is a disaccharide formed by condensation of a glucose molecule and a galactose molecule. All monosaccharides are reducing. All disaccharides except for sucrose are reducing. Glycogen and starch are formed by the condensation of alpha-glucose. Cellulose is formed by the condensation of beta-glucose. Glycogen and starch are both storage molecules with 1 to 4 glycosidic bonds between molecules of alpha-glucose. They also have branches which make it easier for hydrolysis as there are more ends for enzymes to attach. This allows them to be broken down quickly. There are 1 to 6 glycosidic bonds at the branches. Glycogen is the animal storage molecule and so has more branches for faster hydrolysis. Cellulose is a long, straight chain of beta-glucose and is used in cell walls. The cellulose chains are linked together by hydrogen bonds to form strong fibers called cellulose microfibrils. There are only 1 to 4 glycosidic bonds. The Benedict's test can be used to check for reducing sugars. A color change from blue through to red means reducing sugars are present. To test for non-reducing sugars, take a fresh sample of the solution and either heat it with diluted hydrochloric acid or hydrolyze using enzymes. You can then neutralize it with sodium hydrogen carbonate. Once you have done these, you can continue the Benedict's test in the same way you would for a reducing sugar. To test for starch, just add iodine dissolved in potassium iodide solution to the test sample. If there is starch present, the sample changes from brown-orange to a dark blue-black color. In your exams, make sure you always talk about iodine dissolved in potassium iodide and not just iodine alone. Thank you for watching Phreology Revision.